So you go here, you take the sequence and you blast it. Then you know where it comes from. So this is what I have done. I blasted. Yeah, you can look at the peaks, but the peaks will not tell you what they are. It can be. Housekeeping genes would look like this, but you're not 100% sure that they are. If you want to be 100% sure, you're going to blast them. So that's exactly what I did. I blasted them, and what came out? Ribosomal RNA. All of them were perfectly aligned to ribosomal RNA genes. So, why do I have then these very low number of mapped reads? It's ribosomal RNA, so it comes from human. If I map them to the human genome, why don't I find them back? Yeah, it explains why I have a lot of duplication here, what they are, but it doesn't explain yet why they are unmapped. The reason for that is that ribosomal RNA genes are considered repeats. They are not in the FASTA file that you download. And they are also not annotated in the GTF file. So if your mapper is going to map these, well, if he doesn't know that these exist, if he doesn't have the sequence, then he's not going to map against them. So this is why you had such a high number of unmapped reads. And it's not one example. I got multiple examples during the years from people who have this issue. So for RNA-seq data, if you have really low numbers of unmapped reads, the first thing that you should look at is the things that are contaminating here, or the reads that do not want to map. You can choose. Either you go to the FASTQC report, or you go to the unmapped reads, the FASTA file, blast them, and check if they are ribosomal RNA. Where does it come from? Well, you start your library prep with total RNA, and then normally you're going to select the messenger RNA from the total RNA. But it's never perfect, so you always have some ribosomal RNA contamination. But this seems to be really bad, because if you look here, 5% remains. So if you're going to throw away all the duplicates, 5% of these 12 million remain. Now, based on this alone, you don't know if all these things are ribosomal RNAs. There might be housekeeping genes here, and for these housekeeping genes, everything is okay. So you would keep them in, of course. So just based on this, you cannot say how bad it is. It doesn't look good because this is really low, but you have no idea how bad it precisely is. So it is possible that they forgot to clean up the total RNA and to select the messenger RNA, and they just sequenced the total RNA, and that means that 90% of your reads will be ribosomal RNA, because in total RNA, 10% is messenger RNA, 90% is ribosomal RNA. So that means that if you only have 10% left here, that will not work. You can analyze, but it will not give you a lot of info. If it just partly failed, the depletion of ribosomal RNA, you will still have messenger RNA left. And you could proceed with these data, but even there you will know upfront that genes with low expression levels, you will not see them. They will not be detected, because so much of the reads that were sequenced were ribosomal RNA. So, and this is why these ribosomal RNAs, they do not map. So if you have very high levels of unmapped reads for RNA-seq, it's typically ribosomal RNA contamination. So blast them, and then you know. Because there are other reasons why you see high duplication levels. Normally, your sequence provider should do this. The same machine as the one that you use to check if your RNA is degraded will also tell you whether there is a lot of ribosomal RNA in your samples. So if it happens, it means that your provider has not done his job well and you have to complain. You also check duplication, just like you did this morning for the DNA-seq reads. So how can you see duplication? Well, in IGV, if you see a lot of identical copies in IGV, it means you have a lot of duplication. Yeah? You can also look in your FastQC report. You can even look in your QualiMap report. They all have plots that you can use for that. So if it looks like all the reads are duplicated, which is kind of the case here in this upper example, it's typically PCR over amplification. So if you have a small library and you have to do a lot of PCR to get enough fragments, yeah, then you end up risking that each fragment is duplicated at least once. If it's only here and there, then it's the housekeeping genes that you see. So for the housekeeping genes, seeing duplication is okay, but you do not want to see it for all the reads. That's not okay. So if you have really low numbers that remain after deduplication, like here, for instance, yeah, only 5%, then it's not really normal what you see. And what is the problem with the RNA-seq data? Well, the problem in RNA-seq data is that you always have biological duplicates because it's RNA-seq, because the housekeeping genes. If you also have a lot of technical duplicates, you cannot make the distinction between the two, unless you use the UMIs. I explained this last week. The technical duplicates will distort the counts. Your counts will not be accurate. But removing all the duplicates will also distort the counts because you're also removing biological duplicates. So what to do? Well, when it's really bad, and it's not because of ribosomal RNA, but it's because of PCR overamplification, what you see here, the best thing to do is to remove them. So use PICAR, mark duplicates on the RNA-seq data, on the BAM file, even if it's RNA-seq data, and remove them all. If you think that the technical duplicates are worse or are in bigger amounts than the biological duplicates, then it's more accurate to remove them. Yeah? It's not ideal, but it's the best thing that you can do here. Yeah, and then the last thing that you typically check for RNA-seq data is the strand bias. If you haven't done a stranded experiment, then you look here at this one, this RCQC tool, you should see something like this. This is perfect. If you have done a stranded experiment, what do you expect to see? I get a lot of questions about this. So people seem to think in IGV, more specifically, that when you have done a stranded experiment, that you will only see red reads or only blue ones. So that all the reads will map either to the plus strand of the genome or to the minus strand of the genome. And that's not the case. What the reads will do in a stranded experiment is they will all come from the plus strand of the transcripts or from the minus strand of the transcripts. Yeah? But it's relative to the transcripts. So, and some transcripts come from the plus strand in the genome. So these reads will still map to the plus strand in the genome, and some transcripts will come from the minus strand on the genome. And these reads will still map to the minus strand of the genome. So, in a stranded experiment, you still expect to see two colors here. What you don't expect to see in a stranded experiment is a mix. Yeah? Regions where you have both blue and red. That is strange. So, unless it's in overlapping genes, there you will have a mix. But in the regions where there's no overlap between genes, you expect to see only one color. If you have done a stranded experiment, the best way to check is IGV. If you see a mix everywhere, it means, again, your sequence provider has messed things up and he hasn't made a stranded library. A lot of things you check, and I do check them. I check them thoroughly for RNA-seq data because I do want to have my counts as accurate as possible. But that being said, for RNA-seq, the mapping is not that crucial. Yeah? So it's not that you're going to tweak the parameters of star. No, if you get good results, you go on with the results.